Hello friends, welcome back to my channel BioFreak. Let's talk about DNA polymerases part 2. I had already talked about DNA polymerase 1, 2 and 3 in part 1. Now here I'll be talking about DNA polymerase 4 and 5. So this is a para from Pathfinder book. DNA polymerase 4, that is DIN-B, and DNA polymerase 5, that is UD2-C, are Y family DNA polymerases. Now the members of Y family do not contain any 3' to 5' exonuclease activity. That means they do not have any proofreading activity. These polymerases are also characterized by low catalytic efficiency. Okay, then they have this low processivity and a low fidelity. Okay, because they do not have any proofreading activity. They are involved in translesion synthesis and replicate the damaged DNA by bypassing the damaged nucleotide that would otherwise block the normal progression of the replication fork. So basically, Y family DNA polymerases, they are involved in translesion synthesis. Okay, and the second thing is, in translesion synthesis, what actually happens is basically it helps the DNA polymerase to bypass the damaged nucleotide sequence. Okay, because the normal DNA polymerase like DNA polymerase 3 cannot bypass the damaged DNA. In this case, let's look at this particular thymine dimer. Okay, here there is a thymine dimer, and DNA pol 3 for that matter cannot bypass this damage without rectifying it. So here in translation synthesis, DNA 4 and 5 will come, they'll bind to this strand here, DNA pol 3 before will leave, and now this particular DIN-B and UD2-C will come and bind. It will help in bypassing the damage, and it will synthesize the complementary the sequences which may not be exactly like P is not really opposite to G here. Okay, so these are not really the complementary sequences, but somehow it will try to repair it, but it will bypass that thymine dimer. Okay, and later once that damaged nucleotide portion is bypassed, the DNA pol 3 can continue and replicate the further DNA. So that is translation synthesis. Now there are several other repair mechanisms, but in the end when no other repair mechanism is working there, sometimes the DNA is not the damaged DNA is not only always repaired, but sometimes it's overlooked by certain enzymes and then it is bypassed. Okay, so to summarize, Y family DNA polymerases specialize in translation synthesis by passing damaged bases that would otherwise block the normal progression of the replication fork. And Y family polymerases have unique structural features that allow them to bind to the damaged DNA, and these unique structural features are absent in DNA pol 3. Hence its speciality of the Y family polymerases, and it uses its ability to modify the template base to direct the nucleotide incorporation. So that's all about this lecture. Let's look about more DNA polymerases in part 3.